Okay, so last time we talked about prophase and did quite a bit of setup. So let's just remind ourselves what went on there. So it was primarily two events. One was the condensation of the chromosomes began to happen, and two, the centrosomes, and remember these guys would have duplicated back in S phase. They now begin to organize these microtubules in the cell into two star shapes and will begin to push on each other. So most of that is actually going to take place during what the next phase is happening, and that's pro-metaphase. So a couple of things that are going to happen here is that the nuclear envelope obviously has got to disappear, and because remember, the nuclear envelope and the endoplasmic reticulum are all closely connected to each other, so it's going to disappear as well. Now, by disappear doesn't mean it's just taken apart, or I mean that we're getting rid of it. Obviously what's going to happen is it just gets too small. It's going to be taken apart into small little vesicles that can then be transported across those microtubules to opposite ends, where once the chromosomes have been pulled apart, we can essentially reassemble that nuclear envelope and the endoplasmic reticulum on either side in the two daughter cells. All of this is actually going to be taken care of by the nuclear lamina. So remember, that's the structure that lays up under the nuclear envelope in the nucleus and is closely associated with the nuclear matrix. So as the nuclear matrix is collapsing down for the chromosomes, the nuclear lamina is beginning to take apart the nuclear envelope. Once your two sides of the mitotic spindle are formed, then essentially they will have moved to each side, and what we do during prometaphase, or one of the main events that happens during prometaphase, is for those kinetochore microtubules to attach to each kinetochore. And there are going to be mechanisms that ensure that that happens, because think about it, we're about to separate sister chromatids of our chromosomes. We have 46 chromosomes, and we have to make sure that that both sides are attached to all 46 of them. Okay, so a couple of things here. This figure just shows some of what's going on with the nuclear lamina. That would be this green stuff. So as the nuclear matrix is assuming X shapes, the nuclear lamina is disassembling into much smaller structures and is actually going to travel with the chromosomes as they go along. It's microscopic, so you can't see it. And then later in telophase, it will reassemble into the nuclear envelope around the DNA as it gets unwound. Here is some the pictures of the nucleus here, so you can see the little nuclear pores in place, and this would be the little what we call blebbings of the nuclear envelope, and as it assembles over time, it eventually forms the nuclear envelope again. Kinetochore, so remember I told you that the kinetochore is a set of proteins, a complex that actually builds itself around the centromere of one of these chromosomes that's duplicated, and these kinetochore microtubules, and actually you're going to have more than one. I mean, this is, as we pull these things apart, these things are pretty heavy when you think about it, and the microtubules, it's going to take multiple to come in. You can kind of see them here in the picture. So here's the centromere, these would be the arms, and you can kind of see them dragging back. So pulling would be this direction, and here would be the proteins of the kinetochore. These would be attachment proteins, and these would be the microtubules that are attached. And now what's going to happen is, in the next phase, we will begin to pull those apart. All right, so everybody attaches. You'll notice the nuclear envelope is gone, so a couple of things happen in prometaphase. So here you can see that the two different asters are really quite well defined. You can see that they are now attaching to these different chromosomes. The chromosomes are sort of sprayed out a little bit. So for example, this guy has got to reach over and attach here. This one would have to reattach to these. So there's a lot of a lot of movement going on here, and once those are attached, we would be ready to go into the next phase. The next phase is largely just making sure that everything's lined up and ready for division. It's what we call metaphase. And once they have attached, the kinetochores, microtubules, are now going to start to push on the chromosomes. And by both of them pushing, what will happen is it's going to make those chromosomes move to the center. And again, remember that as the two spindles, or the two sides of the spindle, push on each other, it establishes the outer poles of the cell, and the metaphase plate is going to be located near the center of where those are established. When we say pushing, just remember what we talked about with microtubules. Remember that the microtubules get longer or shorter by the centrosome taking subunits in and out. And so in this case, what we have is, remember, the tubulin subunits. Each one of them is going to essentially pull subunits off, add subunits in at different rates, and eventually what will happen is the chromosomes will end up getting lined up. So notice how this guy's got to shorten, whereas this guy has to lengthen in order for the chromosome to get where it needs to be. So one side is always growing, one side is always shrinking until we reach that middle point. And these would be the kinetochore connectors in here, and basically what we're doing is helping things line up. So one of the things you're going to want to definitely look at for lab is when you identify the metaphase. It's a little bit easy to get sidetracked or distracted by the actual arms of the chromosomes. You'll notice that they are not necessarily linked up with the center, but the centromere is definitely linked up, and so this is what defines that metaphase plate. So oftentimes what you'll see in metaphase is something that looks like this, and what you want to imagine here is, yeah, these little tails for the arms of the chromosomes are coming off, but the centromeres are all right here, where the two sides, which are very nicely defined, you can see the astral, and some of these would be kinetochores, some of these would be intrapolar, and now chromosomes are laying right in the middle of what we call the metaphase plate, this zone that's equally distant between each of the two sides. Okay? Alright, so we'll leave it there. That is a good place to stop for this, and we'll do anaphase and telophase in the next section.